opportunity to speak here today. So I'm a postdoc at Stanford on the lab with Catherine Blish, and it's great to be back here at Berkeley, where I did my PhD with Britt Lonsinger, sitting next to Emma, actually. And one of the ideas that Britt imparted to me is that viruses truly know us better than we know ourselves. And from Emma's talk, I think that idea was conserved between both of us. So in keeping with that philosophy, the overarching question motivating my work is how do viruses evade detection by the immune system? And our hope is that by understanding these viral immune evasion tactics, it will shed light on the fundamentals of how the immune system operates during the course of viral infection. This is my Berkeley. I haven't completely become a traitor. So a truly classic example of viral immune evasion comes from influenza A viruses, which accumulate in mutations by a process known as antigenic drift. And this allows it to partially escape from pre-existing antigen-specific antibodies and T cells. Come on, no one's excited about the laser pointer? Okay, it's really weird I was going to get scooped with the laser pointer. So influenza viruses also use antigenic drift, which is the reassortment of a new gene segment giving rise to a new subtype. And these are often the culprits of the periodic flu pandemics that we experience. And the global burden of this disease is immense, 3 to 5 million cases and over 650,000 deaths per year. And these deaths can be partly traced to an overexuberant pro-inflammatory immune response. So while we have a handle on how the immune system, or how flu escapes from the adaptive mode of the immune system, comparatively little is known about escape from innate immunity. And this includes natural killer cells, granulocytes, dendritic cells, etc. So I became particularly interested in the role of natural killer or NK cells for a number of reasons. They're a major immune cell residing in the healthy lung, and they're rapidly recruited post-infection, where they provide an early source of pro-inflammatory cytokines. And data from the mouse model has been really intriguing. A number of reports have found a protective role for NK cells, while others have found that they may contribute to immune-mediated pathology, and this is dependent on viral dose. And in humans, we know that in the last pandemic during 2009, those patients that had severe disease had a stark NK cell deficiency, and this is compared to people with milder symptoms. So how do NK cells work? So instead of becoming activated through those virus-specific receptors like B and T cells, NK cells instead use this truly beautiful array of inhibitory and activating receptors to sense when a cell is infected. And here's a schematic of how these receptors work. Inhibitory receptors on the surface of NK cells recognize HLA class I molecules, and this is basically an all-clear signal, and the NK cells just go along their merry way. However, if these proteins are downregulated, which occurs during infection or cancer, the NK cells then take a second look. And what they're looking for are other warning signals, and these can take the form of activating proteins on the surface of an infected cell or secreted pro-inflammatory cytokines. And if sufficient of these warning signals are detected, NK cells become activated, and they're able to directly kill the infected cell by releasing granules containing perforin and granzyme, or release pro-inflammatory cytokines, and they're famous for their ability to produce interferon gamma. And previous work has shown that flu fails to downregulate HLA class I molecules, but if and how these activating signals are regulated remains unclear. So we asked the very simple question, do flu-infected cells evade NK cell recognition, or do they trigger these NK cell effector functions? So to do this, we harvest PBMCs from healthy blood bank donors. We isolate both NK cells and monocytes, and this is an influenza permissive cell type. And we then co-culture to generate a human ex vivo autologous system to see how influenza manipulates the host cell, which then in turn regulates NK cell function. So we evaluated both killing and interferon gamma production in this study, although I'm going to focus just on the cytokine production here for the sake of time. So we used an intercellular flow cytometry approach, and here are biplots with interferon gamma on the x-axis and cytosine on the right. And we can see that NK cells produce interferon gamma in response to monocytes infected at 7 hours or 24 hours. And this is the pandemic H1N1 2009 strain, which I'll call CAL09. And this is compared to mock-treated monocytes where there's no response. We also had looked at this a number of donors, and one question we were interested in is it really the infected cell that's eliciting this gamma response, or is it the virion, the influenza virion itself? But we can see when we just add virion directly to NK cells, you don't see a response, and you really require that target cell. And the orange is just PMAI, a chemical stimulation, a positive control. So there's really something that's being changed by this target cell by influenza, which then is activating the NK cell. So we next asked if this held true for other flu isolates, and we compared 6 strains dating back to 1946. These are a mixture of subtypes. And we found something a little unexpected. That Cal09 strain, here in blue, led to a markedly higher interferon gamma and NK cell response than all the other strains tested. And at 24 hours post-infection, after several cycles of replication, the NK cell response to Cal09 was preserved. And we've confirmed this in over 40 donors, looked at interferon gamma mRNA, secreted protein levels, and observed this response in influenza-naive lymphocytes as well. And so this raises the question, does this pandemic H1N1 strain lack an NK cell evasion mechanism possessed by the other strains? And if so, what does it consist of? So using an evolutionary distinct flu strain, an H3N2 that circulated around the same time, I'm going to call this VIC11, which does not trigger NK cell gamma protection by 24 hours post-infection, we compared these two strains by transcriptional profiling. So monocytes were either mock-treated or infected with these two strains at carefully matched infection levels. We then harvested RNA from re-isolated monocytes and performed RNA sequencing and differential gene expression analysis using the DE-seq2 pipeline. And here we visualize the differentially expressed transcripts. In Cal09, we have about 162 that were higher. With VIC11, there were 79 that were higher. This is just a classic volcano plot. And a PCA indicates that infection status, which is indicated by color, and time point, which is indicated by shape, had a far greater effect than donor variability. And in full disclosure, I used 3 white men for this study, so that may be part of why that is. Visualization of the cellular transcript heat map revealed that these viruses induce a distinct transcriptional signature. And so to capture the biological significance of these differentially expressed genes, we turned to the string program to construct an interaction network where red are transcripts expressed at higher levels by Cal09. And we observed this module of protein interactions connected by the interferon alpha receptor node. And this includes interferon alpha. So this is indicated of an early transcriptional induction of interferon alpha mRNA, which is now observed by the VIC11 infected cells. So identification of these transcriptional differences prompted us to quantify cytokine protection using Luminex. And here I plotted the log 2-fold change of Cal09 over VIC11. And you can see that Cal09 infected monocytes secreted greater levels of numerous cytokines, including interferon alpha. And this is a known potent NK cell activator. So we therefore asked whether neutralization of interferon alpha in this co-culture impacted the ability of NK cells to produce interferon gamma. And indeed, we saw a significant drop in interferon gamma production by NK cells when you neutralize interferon alpha. And this is in comparison with the isotype control. And interestingly, if you prevent viral replication of these two strains, interferon alpha secretion by VIC11 goes up 10-fold from the dark red to the light red. So this is consistent with Victoria 11 possessing the ability to suppress interferon alpha production, which Cal09 does not possess. So next we asked whether receptor-ligand interactions are additionally required for the NK cell response to flu, or are those cytokines alone sufficient? So to distinguish between these two possibilities, NK cells and monocytes were co-cultured in a transwell system using a porous membrane to separate the cells but allow for cytokine exposure. And under these conditions, we found that elimination of direct contact completely abrogated the percent interferon-positive gamma NK cells. And this is consistent with the requirement of further receptor-ligand interactions. So there's something about the cell surface which is needed. Okay, oops. All right, so does this go the other way? All right, so to identify these ligands that are being changed by influenza strains, we next use cytometry by time of flight, or CyTOF. And this allows for the simultaneous measurement of already 40 cellular parameters on a single cell resolution. And I built and validated a 40 parameter CyTOF panel encompassing antibodies specific to a number of inhibitory and activating ligands. And then we conjugate to rare earth metals, which will then be able to be detected by mass cytometer. So cells from 9 donors were either mock-treated or infected with either influenza strain, MOI of 3, for 24 hours, stained with the ligand panel, and then run through the mass cytometer. So this generates a large amount of high-dimensional data. So to interpret this data, we initiated a collaboration with Christoph Seller in the Department of Statistics. And he built a generalized linear mix model, and this is an extension of logistic regression. And it's able to evaluate which markers are predictive of infection, the Cal09 in blue on the left, or Victoria in red on the right. And this allows for to account for donor-specific heterogeneity. And the top 2 hits were CD112 and CD54, and which are activating ligands that were predictive of Cal09 infection. Another way to visualize this data is to use principal component analysis, where we find the expression of 112 and 54 were the major contributors to barriers between these two strains. Let's see here. So 112 and 54 ligands bind to the NK cell activating receptors 226 and LFA1, and sorry for the immunology alphabet soup. But to test if their signaling through these receptors contributed to the NK cell response, we introduced a blocking antibody, or the isotype control. And we found that blocking these receptors led to a drop in the NK cell interferon gamma response, although the magnitude differs between individuals. So we next analyzed how does influenza modulate these ligands by conventional flow cytometry. And we first assessed 112 and 54 expression on treated monocytes. And here we can see these bimodal populations. And we saw that when we infected with Victoria 11, we saw a marked downregulation of both ligands. And to our surprise, infection with Cal9 also led to a downregulation of both ligands, although not nearly to the same extent. And this pattern was recapitulated on cells from 6 donors, and importantly, this observation was preserved after controlling for the slight differences in infection levels. And finally, we asked whether these downregulation of these ligands occurs on cell lines derived from normal bronchial tracheal epithelial cells. So these are physiologically relevant for influenza. And indeed, both ligands were downregulated, although VIC11 was downregulated to a more robust extent. So now we have this model with differential induction of interferon alpha, and these activating ligands contribute to the magnitude of the NK cell interferon gamma response, although other receptor ligand interactions are almost certainly evolved. So moving forward from here, we're using the influenza reverse genetic system to identify the viral mechanism governing ligand downregulation. And we're also interested in using experimental evolution to identify and characterize flu escape variants. And with that, this work would not have been possible without our collaborators Christoph Seller and his advisor Susan Holmes, the Department of Statistics. I'd like to thank the Giannini Foundation for funding my postdoctoral training. And science is not done in a vacuum, and I feel incredibly grateful to be part of this wonderful lab, and to my advisor, Catherine Blish. And I'll stop there, and I'd love to take any questions if there's time. Questions? I knew Britt was going to ask a question. How do you think the ligands are downregulated? So we've cloned out all of the gene segments and then just done like a 293 transfections individually. And the nuclear protein was the one that came out, but we're still verifying that. It might be enhancing degradation of these proteins. That's one pathway that's been known by other viruses to use, so that would be my top suspect. Yeah. Yeah, I know, HLA is. You have any ideas about why that would be? I mean, it's kind of a marker of other HLA class molecules because of the peptide singers. Yeah, I know, I'm fascinated by that. I was trying to figure out a good experiment to follow up on that, so if you have any ideas. Yeah.
Quick question.
Yeah.
You looked at different age groups because the severity, you mentioned, you commented on so young people, old people.
No, we haven't. All of these blood bank donors are de-identified, so we don't have any demographics on them, unfortunately. Yeah. No questions? Okay. All right. Thank you.